それではお時間になりましたので始めさせていただきます。パセオン株式会社のですね、出展者プレゼンテーションということで、本日はこういったコロナ禍の中ですね、お集まりいただきましてどうもありがとうございます。今日はトピックとしましては、原薬の溶解性、バイオアベラビリティ改善戦略ですね、最適な技術と、あとおよび賦形剤の選択というところで、プレゼンテーションを始めさせていただきます。あと今日のプレゼンテーションなんですけれども、アメリカにおりますですね、技術者による事前に録音されたプレゼンテーションとなりますので、あらかじめご了承いただければと思います。よろしくお願いいたします。まず私の方から最初にですね、申し遅れました。私事業開発の谷本と申します。まず最初に私の方から会社概要を簡単にご案内いたします。まずサーモフィッシャー、我々の親会社のサーモフィッシャーサイエンティフィックという会社ですけれども、世界をリードするサイエンス企業としまして、サイエンスサービス企業ということで、現在8万人ほど従業員を抱えております。そのうち約5700名ほどがサイエンティスト、エンジニアになりまして、年間のですね、研究開発の投資額が約10億ドルということで、約1000億円ほどですかね、毎年投資、設備、研究開発に投資をしております。売上額がですね、300億ドルということで、大体日本円で3兆円の規模となっております。で我々サーモフィッシャーのですね、ミッションなんですけれども、私たちのこの住む世界をですね、より健康で清潔、そしてまた安全な場所にするためにサービスや製品をご提供しております。でサーモフィッシャーサイエンティフィックの中にはですね、大きく6つの主要ブランドがありましてですね、左から分析関係であったりとか、遺伝子の解析であったりとか、あとはラボ関係の消耗品ですね、そういったものを取り扱っていて、で今回ご案内する我々の CDMO のサービスにつきましては、一番右にあたるパセオンですね、こちらが医薬品の開発製造の自宅サービスということで、パセオンブランドとして展開しております。パセオンなんですけれども、業界で唯一のエンドツーエンド CDMO サービスをご提供しておりましてですね、こちらの左の円グラフですね、我々のサービスの中心になりますのが患者様ですね、あと患者様と、あとは我々のお客様であります医薬品メーカー様、バイオベンチャー様であったりとか、そういった患者様を中心にですね、右上から原薬製剤の開発のサービスから、この辺は前臨床の段階からお手伝いをしておりまして、で実際に動物試験や臨床試験向けの治験薬製造ですね、こちらは原薬と製剤ともにサポート可能です。あとは治験薬の包装ラベリングですねに加えまして、治験薬の保管配送、ここもエンドツーエンドでサービスをご提供しております。あと実際に製品が承認された後ですね、原薬製剤の商業生産の手伝いもしておりまして、あと商用向けの包装ですねもご対応しております。あとはライフサイクルマネジメントの一環としまして、ジェネリックやバイオシミラーの開発のお手伝いもしておりまして、あと近年ですね、細胞遺伝子治療、セル&ジンセラピーの領域に参入いたしまして、特にウイルスベクターであったりとか、プラスミドの DNA ですね、こういった遺伝子細胞治療に向けた CDMO のサービスをご提供しております。こちらがパセオのサービスの、いわゆるエンドツーエンドサービスですね。で現在ですね、約1000社ほどのお客様との付き合いがありまして、もちろんトップ20に入るメガファーマ様との長い実績もあるんですけれども、約8割近くのお客様がですね、いわゆるバイオベンチャー、スペシャルティファーマのお客様に当たります。でこれまでですね、過去10年間で109品目の製品承認実績がありまして、2019年に関しましては6000件以上の治験プロジェクトに携わりました。あと現在、グローバルで3500名ほどのサイエンティストを抱えておりまして、これまで80カ国以上のお客様に開発、製造の支援をさせていただきました。で現在ですね、我々55施設を保有しておりまして、いわゆる治験薬の開発、製造、製剤の開発であったりとか、原薬等々ですね、55施設グローバルに展開しております。でこういったキャパシティからですね、現在約1日に100万人ほどの患者様に治験薬や医薬品をお届けしております。で今日ご案内しますですね、プレゼンテーションにつきましては、我々の経口固形製剤の施設から のご対応となりまして、いわゆる我々特にアメリカ、ヨーロッパにですね、約 10施設、経口固形剤の施設を保有しておりますが、本日の、溶解性改善のサービスにつきましては、このベンド、アメリカのベンドの施設からでご対応しております。以上ですね。これから本題のプレゼンテーションに切り替えさせていただきます。少々お待ちください。じゃあこれから開始させていただきます。
This is an overview of Patheon's Quadrant 2, an innovative approach for solubility and bioavailability enhancement. It is well known that in today's environment is increasingly complex with increasing risks. It is estimated that 70 to 90% of molecules have solubility and bioavailability challenges. And there's a lot of iteration at each phase of product development, and the probability of taking a poorly soluble molecule, an NCE, to be a commercial product is increasingly risky and increasingly complex environment. So a traditional approach to solubility enhancement is a trial and error approach similar to this ball bouncing around in a maze. There can be a lot of dead ends and missteps at each stage, and in our analysis the duration to get to phase 1 CTM takes about 14 to 19 months. With the Quadrant 2 approach, it's a much more streamlined approach, and we have eliminated the roadblocks and the dead ends, and we can get an NCE to the clinic in a much faster manner. And in our analysis, the duration to get to phase 1 CTM takes about half the time, 8 to 9 months. So how does Quadrant 2 work? With Quadrant 2, we take the API chemical structure and its physicochemical properties, and of course business and clinical objectives are important, and we input them into our computational algorithms and models that we have developed here at Patheon Thermo Fisher, and the output is selection of the best technologies as well as formulation, excipient selection. So we input the structure and the properties of the API into our computer models, and Quadrant 2 can predict the technology and excipient selection for formulation. So a deeper dive into how this works: once we input the structure and the properties of the API into our selection algorithm, the output is like a traffic signal: green, yellow, red. Green implying a high likelihood of success. Yellow means likely to succeed, but it would require further experimentation. Red implies most likely not going to succeed. Some of the logic under the hood: for example, we often see APIs which are really poorly soluble, with a solubility of less than 1 microgram per ml, and very high melting points, say for example 300 Celsius. And for such APIs, things like hot melt extrusion will not work due to the high melting point, and simple size reduction will not work due to the high crystal lattice energy. And we bracket this as a function of dose: low, medium, high. Low dose is less than 50 milligrams, medium is 50 to 200 milligrams, and high dose is greater than 200 milligrams. And this gives you a starting point, a roadmap for selection of the best technologies for a poorly soluble compound. The next step of Quadrant 2 is the excipient selection process. We again take the API chemical structure and its physicochemical properties, input it into the Quadrant 2 program, and the output is selection of the best excipients for formulation. The reason excipient selection is important is when it comes to amorphous dispersions, there's a wide variety of chemistries to choose from. As you can see on the slide, for a amorphous dispersion, whether it's spray drying or hot melt extrusion, you have cellulosic polymers, polymethacrylate polymers, polyvinyl polymers, and once you start adding additives like surfactants, permeation enhancers, it can get very complicated. By determining the best polymer empirically, it can be time consuming and also resource consuming. So what we do with Quadrant 2 is we run computer simulation of drug and these excipients and select the best excipients for formulation. And we narrow down the list to the top 5 or 6 to do experimental screening. So all this is done computationally by Quadrant. A little more deeper dive into what kind of modeling we do for excipient selection. We basically take the API and assign different descriptors, and we look at things like potential energy, charge surface, potential energy bonding surface, things like hydrogen bond donors, acceptors, aromatic, hydrophobic. We run a series of quantum calculations as well as molecular dynamic simulations in order to select the best excipient for any given. We do the same thing for these different polymers like we do for the API, and we essentially try to select the best polymers for formulation by doing this computational modeling approach using Quadrant. I mentioned molecular dynamic simulation. This is an example of a molecular dynamic simulation for a poorly soluble compound, dipyridamole, which is shown in the gray space filling model, and the polymer PBAP, which is shown in the red and the blue, and we get insights into the interaction between the drug and the polymer, and we can rank order the best excipients for formulation moving forward. So there's a very rapid way we can computationally screen different polymers by running these simulations and help in the formulation of the amorphous dispersion. We also get insight into maximum drug loading. Here I'm showing you for a spray dried amorphous dispersion of dipyridamole, which is shown in the ball and stick, and the polymer is HPMC-ASM. We are looking at potential drug-drug interactions as you get up in drug loading. As you can see, as you increase the drug loading, once you get over 48% drug loading, you will start getting interactions between the drug and the polymer, and this can have an impact on physical stability. So by running these molecular dynamic calculations, we can get prior insight into how much drug loading and how much physical stability one can achieve by formulating these spray dried amorphous dispersions. This can also apply to hot melt extrusion as well. So the Quadrant 2 technology and excipient selection has a proven accuracy. We have validated this with over 250 molecules. As you can see, the accuracy of our technology selection algorithm is over 90%, and the accuracy of our excipient selection algorithm is over 80%. Some examples from the literature are shown on the top. Posaconazole, Noxafil, we predicted an HME approach would work. The commercial product Noxafil marketed by Merck is an HME. In the middle is a drug called Roquetrol, and we predicted that a lipid-based approach would work, and the commercial product by Roche is a soft gel. And on the right-hand side, itraconazole, we predicted that a coated bead approach would work, and Sporonox, marketed by Janssen, is a amorphous dispersion in a coated bead. And we also predicted hot melt extrusion would work, and life cycle management of itraconazole recently got marketed as Onmel as an HME product. Getting started is quick and easy with the API chemical structure and its properties. The subject matter experts will conduct the analysis in 2 weeks and will provide a review of our findings, followed by a comprehensive report with the path forward. And this is a very rapid way to select technologies as well as formulations in a very cost-effective manner. Where does it fit in the overall approach to accelerate the timeline to the clinic? So as you can see from this slide, the computational modeling takes 2 weeks. Preclinical development will take the next 6 to 8 weeks. And what we do is feasibility studies and PK supplies. And after that, once you get PK results, we will work on the dosage form, the drug product, formulation, process development, and scale up, which will take the next 4 to 6 months, resulting in the first-in-human studies, supplies for the first-in-human studies. So summarizing, Quadrant 2 is part of an integrated process to get your molecule to the clinic fast. Our motto is work smarter and faster. So we have developed these exclusive computational models that are scientifically based and are validated and highly accurate. And we have the expertise and broad formulation capabilities to take your molecule quickly to the clinic. And we have a global network with a proven record of approval. We are agnostic to the selection of technologies. We have the capabilities to address all of these challenges when it comes to solubility. We can support you in spray drying, hot melt extrusion, coated beads, size reduction, lipid-based approaches, and complexes. We want to help your molecule get the best possible technology for success in the clinic when it comes to bioavailability. So to summarize again, our predictive molecular modeling, Quadrant 2, is helpful for technology selection and excipient selection. We do a series of calculations that I mentioned that involve things like quantum mechanical calculations, descriptors for the API, and charge analysis for the API and the excipients. We can do the BCS, DCS assignment and help in selection of the technology. We run things like molecular dynamic calculations, and with our database of excipients, we can provide you a comprehensive report with the path forward in as quick as 2 weeks. So we can support you from preclinical, animal PK, phase 1 to late stage clinical and commercial scale as well. Here's a case study in which we did predictive modeling for a client for an NCE selection, reducing solubility risk for lead discovery compounds going into preclinical assessment. The challenge was the client had 7 NCE leads with high activity target, so which leads to advance for tox batch analysis, synthesis, and preclinical assessment. The risk was that very low aqueous solubility was observed and poor PK exposure was expected. So we provided a solution with Quadrant 2. We did a computational study on 3 NCEs from a wide range of chemical space in 2 weeks, and we helped in selection of the technology as well as excipients, and we reported the BCS, DCS class as well as different technologies that would help in improving solubility. We found lipids were suitable for 2 NCEs, amorphous spray dry dispersion for the 3rd NCE, and we helped in excipient selection for both lipids and amorphous dispersions. So the result was we moved quickly from the medchem route to the tox batch synthesis for the lipid soluble NCE, and we are getting ready for formulation development for the preclinical supply for the animal tox study. Here's how a snapshot of the Quadrant 2 analysis looks like. By inputting the structure and the properties, we can present you the biopharmaceutical classification system. We look at the conformer quantum mechanical calculations on the top right, the descriptors that I mentioned before, things like the hydrogen bond donors, acceptors, aromatic hydrophobic, potential energy, bonding surface. The surface charge model is shown on the bottom left, and molecular dynamic simulation looking at drug polymer interaction is shown on the bottom right. So these are the types of calculations and presentation that we provide for your molecule. This is the part of the quadrant 2. And next, I'm going to switch gears and talk about spray drying and our integrated offering solutions. We have global capabilities when it comes to spray drying. We can support you from preclinical to commercial scale up. And we have integrated drug substance and drug product services with a proven track record for solubility enhancement and other applications. So a snapshot of the spray drying from preclinical to commercialization. We can handle low to highly potent compounds. We can handle grams to metric tons. And as I mentioned, we have analyzed over 250 molecules using quadrant 2. Over 130 molecules have been spray dried, and over 75 molecules have progressed to the next development phase. We can scale up from development to the commercial scale and integration of API, spray drying, and drug product services. The early development sites are in Bend in Oregon, USA, and Milton Park in the United Kingdom. In Bend and Milton Park, we can do non-GMP as well as preclinical leading up to phase 2B. We can support you in full formulation and analytical support for the drug product as well as spray drying. And we leverage quadrant 2 in the initial stages, as I discussed, to select the polymer selection when it comes to spray drying. The late stage and commercial capabilities are in Florence and Cork. So phase 3 registration through commercialization can be done in Florence and Cork. We use QED approaches for for process development. We have PAT tools and full analytical support. And we have a robust technology transfer pathway from the early development sites to the commercial sites. We understand scale up, process validation very well, and we can help you support in a successful technology transfer, and as well as an integrated drug substance, drug product offering. So the map for spray drying is shown in the slide. As I mentioned, the preclinical phase 1, phase 2, the Bend, Oregon in USA, and Milton Park in the UK can support programs. And Florence in South Carolina has phase 3 and commercial spray drying. Cork in Ireland can support phase 3 commercial. And I must mention the Cork facility can only support aqueous spray drying, whereas the other spray dryers in the network can do organic solvents very routine. So the spray drying equipment scale, some more detail I'm showing you on the slide from the preclinical to commercial, from the left to right. The preclinical, we have Buki B290s in Bend and Milton Park for supporting phase 1 and phase 2. We have an anhydro MS35 in Milton Park and a GEA mobile miner and anhydro MS150 in Bend, Oregon. So here we can support hundreds of grams to kilograms to tens of kilogram scale that can easily support up to phase 2B. And when it comes to phase 3 and commercial, we have an anhydro MS400 in Florence and GEA PST3 in Cork, which is aqueous only. And we are ready to install and qualify an anhydro MS1250 in Florence in end of 2021. And that can support you in large commercial quantities, hundreds of kilogram to metric ton scale. So again, summarizing our network, we have various centers of excellence and one global network. The early development sites are in Bend, Oregon, and Milton Park in the UK, as I mentioned, when it comes to poorly soluble compounds. And then we have the larger sites in the network in Toronto in Canada, and Whitby and Cincinnati in Ohio. We can support you in drug product all the way through commercial. We have soft gel sites in High Point, North Carolina, and Tilburg in the Netherlands that can help you in soft gel formulation, process development, and commercial products. And we have a drug product site in Europe that can support mid and late stage OSD in Bourgoin in France. And we have commercial spray drying in Florence, South Carolina, as well as in Cork, Ireland. So as you can see from this map, we have a wide network with centers of excellence in different sites, and we can support your product starting from all the way from API manufacturing through making enabling formulations for improving solubility, as well as manufacturing drug product all the way through commercial manufacturing. And along with Fisher Clinical Services, we can also support you in clinical packaging and distribution. So with that, I conclude my talk, and please feel free to reach out to the Thermo Fisher team at CPHI Japan in 2021, and we're happy to support your molecule when it comes to improving solubility and bioavailability.
はい、ご清聴どうもありがとうございました。本日ご案内しましたですね、クワドラント2サービスは、実際に原薬を使わずにですね、コンピューターベースでですね、インシリコモデリングで、約2週間で解析をするという、画期的な、しかも手軽なサービスとなっておりますので、その御社の開発品目の中で軟水溶性化合物にあたるものがありましたらですね、ぜひお気軽にですね、ご相談いただければと思います。以上をもちましてですね、パセオン株式会社の出展したプレゼンテーションを終了させていただきます。どうもありがとうございました。